Hi, my name is Miss Dolney. I'm a biology teacher here at Haltom High School. And today we're going to discuss microscopes. We're going to talk about two main concepts when it comes to microscopes. We're going to talk about what are microscopes used for and how do we actually use a microscope appropriately in the lab setting. So the first thing we need to discuss is what are microscopes used for. The word part micro means small, so we use microscopes to see small things. Things like a cell. That could be bone cell, it could be muscle cell, it could be brain cells, it could be cheek cells. We can also use microscopes to see unicellular organisms, which are living things that are one cell big, if you will. Their whole being is just one cell. So we're going to use microscopes to see things and investigate things in the lab that we wouldn't be able to see just using our eyes, or any other tools. Okay, the next thing we need to discuss are the parts of the microscope. So to start off, microscopes have an on-off switch to turn it on and to turn it off. Microscopes have an eyepiece. This is where you actually put your eye to check things out, okay? The microscope eyepiece actually magnifies whatever item you're looking at 10 times. So your magnification and making things bigger already starts in your eyepiece. If you'll notice, you'll go down here, these are called your objective lenses. These lenses are what actually magnify it even more. They make your specimen, whatever you're looking at, even bigger. It goes from 4 to 10 to 40. But remember, your eyepiece also magnifies it 10 times. So really, to find your magnification, you'll take whatever number is here and multiply it by 10. So for example, the shortest one is 4, but we multiply it by 10 because our eyepiece magnifies it by 10 times. And so it's actually a magnification of 40. So it's taking whatever you're looking at and making it 40 times bigger. You'll always start on this smaller objective lens, but we'll talk about that in just a moment. This part right here is called the stage. That's where you'll actually put your slide that you are investigating, and you hold it in place with these little clips. Down here, you'll see two knobs. I see a lot of students sometimes confuse these two knobs, which can cause some problems. Okay, this first knob is called the coarse adjustment knob. It moves the whole stage up and down. You'll use this at the very beginning of setting up your microscope, and then you won't use it much after that. The smaller one is called the fine adjustment knob. A lot of times when you look at a specimen under the microscope, it looks fuzzy or not very clear. That's when you would use this knob to make it crisper and clearer so that your eyes can see it. So if you're sharing a microscope with people in the classroom, you might have to... Coach Fears, everyone.
Hi!
You might have to adjust this based off of your eyes. Sometimes it changes. Okay, for our last section, we need to talk about how to actually use a microscope. And for this, I brought in the wonderful, the lovely Coach Fears.
Hi guys, what's up?
So Coach Fears is going to do the steps of setting up a microscope as I describe it. So, first things first, you have to turn the microscope on, which I've already done for Coach Fears, so that the light can actually shine and you can actually see what you're doing. The next thing you're going to do is make sure that the objective is on the shortest magnification. So it already is there. Lovely. So that you don't hit it because you could break it, and that would be terrible. You're going to move the stage all the way down. Oh, there you go.
There we go.
Then you're going to actually put your specimen onto the stage. So if you'll notice, Coach Fears has her slide. What is it, Coach Fears?
This is a cardiac muscle.
Heart. Thump, 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 thump.
Of a human, by the way.
Okay, so she's going to slide it onto the stage, and she's going to clip the
Excuse me.
slide under the clips. So step 1, turn it on. Step 2, move the stage down. Step 3, make sure you're on the lowest objective. It should have a 4 on it. Step 4, put your slide in place. So Coach Fears has now done that. Coach Fears, what do you notice about the actual specimen? Where is it? Is it off to the side?
Oh, no, it's in the center of the circle.
Very nice. So there's a circle where the light comes through. You need to make sure that your specimen, which is usually a pink or a blue color, is right over it so you can actually see your specimen. All right. The next thing you'll do is move your stage all the way up. Or bring it into focus. So Coach Fears is going to look into the eyepiece, and she's going to bring it up until she thinks it is just about in focus.
Oh, yeah.
Oh?
Yep.
Looks like she found
There
it.
it is.
So you're going to begin to focus. So the next step is to begin to focus using the coarse adjustment knob first. And if you remember, that's the big knob. So once Coach Fears has got it in place, she's going to use the fine adjustment knob, the small one, to make it as clear as possible for her to look at. Just say when, Coach Fears.
You have coffee breath.
Thank you.
Oh, there we go.
So do you.
Thank you.
So, let's review the last couple steps of how to use a microscope. So, step 1, turn it on. And make sure it's plugged in. Step 2, move the stage all the way down. Step 3, put the objective on the lowest power. Step 4, put your slide in place. Step 5, move your coarse adjustment knob to bring the stage up. What number are we on?
6.
Step 6, use your fine adjustment knob to make the image as clear as possible. Once you've done that, you can move to the next highest objective. But do so carefully so that you do not damage the objective on the stage.